So I hope that you have sent questions from all of you from this interesting area of knowledge. So we have in here a couple of questions already, and one is for Jack. It says, what are your thoughts on applying artificial intelligence to hematological neoplasms? Some pros, some cons, any specific limitation?
I think that it'll have similar limitations to looking at tissue, is that namely how do you generate that ground truth? And so I think the manual annotation tends to be very tedious. I think that there probably are lab techniques to accelerate that. There may also be other techniques where you utilize pre-trained networks to pre-label things. The whole limitation just really comes down to the amount of labor it takes to generate that labeled training data. So not the images itself, but the label that says what this is, and that has to be done by a human. So I think that the general goal is you can't get the human out of the equation, but what you're trying to do is accelerate it. So I think that there's some similarities to tissue that you could exploit, but I think that you'd probably still be looking at some new technique development, but new technique development that had promise.
Thank you. So we have a couple of questions regarding the microbiome, and for Jeremiah, and one is how diet, probiotics, or other common over-the-counter drugs may affect the microbiome and the response that they may have to specific therapies.
Yeah, that's a great question. I don't know. I think we had a speaker here yesterday from Cold Spring Harbor that was talking about diet and the microbiome, showed pretty convincing data that diet can have an influence on cancer. But we do quite a bit of work with showing that the diet influences the microbiome, which influences the immune system and inflammatory bowel disease, but we haven't looked in cancer. But I think it's another environmental factor that we're all participating in that we should look in more.
And maybe related to that, another question says how useful it could be or feasible to design a specific microbiomes to boost the immune system in a specific patient that may be already quite advanced or with compromised immune systems.
Yeah, that I think is much more practical. So you might know that Mount Sinai is one of the largest fecal transplant programs in this area. They do about 60 fecal transplants a year for C. difficile. And the same type of idea has been somewhat beneficial in the context of inflammatory bowel disease. So there's a number of companies trying to develop cocktails of microbes that boost the immune system in different ways to try to treat immuno diseases. But I think what we really need right now is some basic science discoveries to make sure we're careful about doing this in a way that we know the mechanisms and we know what our goal is, and not just sort of throwing microbes around. So I think it's a good idea. It has a lot of potential, but we need to really figure out some of the details.
Thank you. And for Christine, we have a question here relating if you are developing the capacity for spatial single-cell sequencing, or where you see that area going into.
That's actually a great question as well. It's definitely something that we've been very interested in pursuing. We have some thoughts that we're starting to kick around. If anybody has ideas of specific approaches that they'd like to try or specific applications that they'd like to see it demonstrated in, that would be great. But that's definitely a particular interest of mine that we're going to be advancing soon.
Terrific. Do you think that with the technology that we have, it will allow you to do that? Any new developments that we need there or resources?
I think with combinations of existing technology, maybe in sort of creative ways, we could definitely get there.
Terrific. So another question about the microbiome, and the question is very specific. Does the microbiome change with age? Is any experimental data in animals maybe being acquainted that can address that?
Yeah, so we know it changes dramatically from 0 to 3. And then from 3 to quite old, it doesn't really change too much. I mean, it's certainly changing, but that's the most stable period. And then I don't know if there's anything published, but there's definitely hints that like 80s, 90s, when you get quite sort of octogenarian, it starts to change again. So definitely changing with age, but for the bulk of your life, it's quite stable.
A question related to the beacon system, and says that rare single-cell isolation, such as circulating tumor cells or disseminated cells in lymph nodes are sometimes difficult to extract and to analyze. Do you think that this application may help, and what would be the throughput that one would be able to expect from that?
Sorry, I missed the first part of the question. Could you
So the
repeat it?
first
Yeah.
part is how Beacon may help us in the context of isolating or putting
Okay.
together single cells,
Gotcha.
such as what we are doing with circulating tumor cells or rare subpopulations that you can isolate from lymph nodes, bone marrow.
So that's a great question. Obviously, sort of the output that we get from Beacon. So Beacon is fantastic at, it's basically a single cell handling technology. So you have all of these pens, you can move various cells around, but the output of your data is going to definitely be dependent on how good your sample is going in. Having said that, we're getting very good at figuring out what the quirks are and what we need to do in order to optimize that. And we've started putting some very unusual samples on Beacon to test the limits. One advantage to our relationship with Berkeley Lights is that we have a lot of leeway in terms of how well we can hack the system. So there's a lot of stuff that we've been doing to optimize that. We actually just did an experiment, Ying just left, but we did an experiment yesterday putting organoids on the chip, on our chips, which is something that you wouldn't think we could normally do, but it's definitely, you know, understanding the system really helps. So that's something that we're working towards. We're analyzing more challenging samples that are harder to get on the tool.
Very good, thank you. Another question related also to the microbiome and the composition, and maybe that can be addressed by different speakers, which is, has been any publication correlating microbiome composition with a specific genetic markers or predisposition factors or signs of indication of early diagnosis or staging of a disease?
Yeah, so far there's been a huge number of studies showing that there's sort of associations between the composition of the microbiome and the disease status of the host. Those have been always this sort of chicken and egg. Is the microbiome responding to the disease, or is it actually different? And most of the time when you then repeat this kind of work in animal models, before the animal gets sick, the microbiome looks normal, and so it looks like it's having more of as a response. There's also been work on doing sort of GWAS and trying to associate genetic markers in people to see if those select for the microbiome. Those have been, there's been 2 or 3 positive studies there, but nothing super significant. So the microbes that have been associated with your genetics have always been more rare microbes that not a lot of people have. Or for example, about a quarter of people have methanogens inside their gut, and those are more likely to be shared between identical twins. But still, not really strong associations between your genetics. It's more your association with the people you live close to. So siblings, for example, share more microbes than random strangers.
So we have just an incoming question for Uri, and it deals with the accuracy of phage-based methods and how you can address the non-specificity of some of the binding receptors or molecules as a potential problem that can interface with the assay. So the first question is the accuracy, and the second, the non-specific binding.
So the accuracy is not as much the problem with the assay as is the sensitivity, where because the peptides you're displaying are actually short peptides rather than full-length proteins, you're actually potentially missing a large number of real epitopes. But other than that, the accuracy should be totally fine. In fact, the assay is a really good platform potentially for specifically studying things like cross-reactivity, because you can actually build a library that contains a large number of variants of a particular antigen of interest, and take particular antibody samples and see, essentially quantify exactly how cross-reactive they are to different variants of the same protein, for example.
Thank you. And for Sasha, a question regarding how the Human Immune Monitoring Center can help in putting together some of the different techniques that you're offering and resources, sometimes to attack a specific problem. Would you be able to integrate the different platforms?
And obviously that's the hardest challenge to do, because you're dealing with lots of data, and so how do you integrate them all? And we do have tools that I didn't have really a chance to show, to at least in a heat map format try to put together data from different data sets and see whether they can cluster in some ways, but that requires also finding potentially common denominators throughout these data files. So like a vocabulary of like, what is a CD8 cell within the IHC data set versus the cyto data set versus genomic data set, and can we put them together and see whether there's something that shows up and clusters. So these are the type of approaches we're taking right now, but it's obviously requires potentially more bioinformatic tools to try to integrate.
Terrific. Thank you very much. Well, we are almost off time, but we are still on time. So thank you to the panel for a wonderful session.